Hello, welcome to this channel. Today we are going to understand the effects of glucose level, cyclic AMP and cAMP on Lac Operon. I will also discuss the consequences of mutation in different Lac genes or the genes of Lac Operon. You can consider this video as a second part of my previous video on Lac Operon. For more details about the Lac Operon, when it will switch on and when it will switch off, you can check my video on Lac Operon. Now let's first refresh quickly our knowledge or what we have understand from my previous video on Lac Operon. If you did not check this video, then I request you to first go through this video on Lac Operon. Now in Lac Operon, when it remains in switch on mode, from the regulator gene, the mRNA will be synthesized, and from this mRNA, the repressor proteins will be synthesized by translation. This blue color box are actually the repressor protein. Now since here lactose is available to the medium, so from the lactose, its isomer allolactose will be produced, and this allolactose, as the repressor protein has a binding site for allolactose, this allolactose will bind to this repressor protein. These blue color balls, these red color balls are actually the allolactose, and blue color box are actually the repressor protein. So allolactose is available and it will bind to the repressor protein, and this causes conformational change in the repressor protein, and as a result the tetramer will not be produced. And since there is no tetramer, there is nobody to block this operator site. Operator site is free. As a result, when RNA polymerase will bind to the promoter, it can slide over to the structural genes Z, Y and A to perform their transcription and synthesize a polycystonic mRNA. And from this polycystonic mRNA, 3 polypeptides or specifically enzymes, namely galactosidase, permease and transacetylase are synthesized, and these enzymes or proteins in turn performs the transport and metabolism of lactose in bacteria. But is there any other molecule which also plays vital role in turning on this Lac Operon? Yes, the other molecule is CAP, catabolite activator protein. It's a dimeric protein, and it generally binds to this CAP binding site, this yellow color box, CAP binding site. Why it is so important? As if CAP is not present, then this RNA polymerase enzyme, which is necessary for transcription, cannot bind to this promoter more efficiently. However, this CAP alone cannot bind to this CAP binding site. It requires the help of another molecule called cyclic AMP or cAMP. When cAMP and CAP binds with each other to form a CAP-cAMP complex, then only this complex will bind to this CAP binding site, and thereby induces the binding of RNAP to the promoter site more efficiently. Thus one thing is clear, the CAP is actually an activator of Lac Operon, as the binding efficiency of RNAP to promoter increases when the CAP is present, or when CAP along with cAMP binds to this CAP binding site. Now you may ask about the exact location of CAP. In some operon, for example in Lac Operon, this CAP region is actually located around 61 nucleotide upstream of this core promoter. It located in the upstream region of core promoter. But there are some operon where the CAP region and the core promoter region overlap with each other. Like for example, say if this is the region of CAP, then some portion of CAP is actually also the portion of core promoter. So this is actually the overlapping region. So in some operon, the CAP and core promoter overlap with each other, and in some operon, the CAP is located upstream of the promoter region. Now this CAP is actually, or CAP binding site, I am talking about actually this CAP binding site. This CAP binding site is actually a 22 nucleotide long sequence, and it contains a consensus sequence of T, G, T, G and A. What is consensus sequence? Consensus sequence is a kind of DNA sequence that remains same and also performs the same function in different organisms. So I think now the concept of CAP and the binding site of this CAP molecule, when CAP will bind along with the cAMP, and how it stimulates the binding of or induces the binding of RNAP to the promoter to initiate the transcription of these structural genes necessary for the metabolism of lactose is clear to us. However, apart from this cyclic AMP and catabolite activator protein, commonly called CAP, another molecule, glucose, also plays vital role. Let's see how. First, we know that in absence of CAP, the Lac promoter is a weak promoter. It means that if CAP is absent, the binding efficiency of RNA polymerase to promoter is low, and hence there will be low level of transcription. Second, binding of CAP to CAP binding site requires cyclic AMP. It means that if cAMP is absent, the CAP alone cannot bind to CAP binding site to allow the binding of RNAP to promoter. Now comes the role of glucose. Glucose directly controls the level of cAMP inside the cell. If glucose level is high, it will inhibit the synthesis of cAMP and also lowers the level of cAMP by efflux mechanism. It induces the outflow of cAMP from the cell. Thus, for efficient binding of RNAP to the promoter to initiate the transcription, the bacteria must need cAP, that is CAP. Without CAP, it will not, the RNA polymerase will not be able to bind to the promoter. Along with this CAP, the bacteria also need a high level of cAMP, because without cAMP, the CAP alone cannot bind to the CAP binding complex. And third, the level of glucose must be low, because if there is a high level of glucose, there will be low level of cAMP, and this in turn inhibits the binding of RNAP to the promoter to initiate the transcription. Thus, for efficient binding of RNAP to the promoter, the level of glucose must be low. or the level of cyclic AMP must be high and the CAP must also be present. Let's see in the figure. In the first figure, we can see there is a low level of glucose. So we know glucose and cyclic AMP are inversely proportional. If there is a low level of glucose, there will be a high level of cyclic AMP, and since there is a high level of cAMP, the CAP cyclic AMP complex will form and it will bind to the CAP binding site, and as a result the RNA polymerase will bind to the promoter site more efficiently. Now the second condition is also fulfilled here. The lactose is present here. If lactose is present, its isomer allolactose will form, and this allolactose will inhibit the repressor, and as a result the operator site is also free since the repressor is inhibited by the allolactose. So in the first figure we can see both the conditions are fulfilled. The operator site is free as the lac repressor is inhibited by the lactose or allolactose, and second, CAP and cyclic AMP complex is present, which allows the efficient binding of RNAP to the promoter. Now move to the second case. Here there is a high level of glucose, and because of this high level of glucose there will be a low level of cyclic AMP, and since there is a low level of cyclic AMP, CAP and cAMP complex will not form, and in absence of this complex the RNA polymerase will not be able to bind to the promoter site. Okay, so here the first condition is not fulfilled. RNA polymerase will not bind to the promoter efficiently because the cyclic AMP is low. Whereas the second condition is fulfilled. Lactose is present, so allolactose will form and it will inhibit the lac repressor, so operator site is free. But though the operator site is free, the RNA polymerase is not binding here because of the absence of cAMP. Okay, so for efficient transcription, the operator site must be free and the RNA polymerase must bind to promoter. The RNA polymerase will bind to the promoter only when cyclic AMP and CAP complex is present, and this occur only when the level of cyclic AMP inside the cell is high or the level of glucose is inside the cell low. Thus, for strong induction or high level of transcription of the structural genes of Lac Operon, these two conditions must be fulfilled. Number one, the allolactose must be present, which will inhibit the repressor and keeps the operator site free for the passage of RNA polymerase, and the second one is the level of glucose must be low so that there will be a high level of cyclic AMP, and this will form a complex with CAP, and this in turn will bind to the CAP binding site, hereby allowing the binding of RNAP to the promoter to initiate the transcription of structural genes. So this is now clear. I think the level of cyclic AMP must be high, and this is only possible when the level of glucose is low, and the second one, the allolactose must be present because it will inhibit the repressor and keeps the operator site free for the passage of RNA polymerase. Now let's understand this interplay of glucose and cyclic AMP through some examples. Till now we have understood that for high level of transcription, the lactose must be present in the medium to inhibit the repressor and keep the operator site free, and second, either the level of cyclic AMP must be high, or in other words, we can say the level of glucose must be low. So in the first case, see, the level of glucose is low here, whereas the level of cyclic AMP is high. So the second condition is fulfilled, means the level of cyclic AMP is high here. So the RNA polymerase can bind to the promoter. Along with cyclic AMP and CAP, the RNA polymerase can efficiently bind to the promoter to initiate the transcription. But you can see the level of lactose is low here, and because of the low level of lactose, the Lac repressor cannot be blocked, and the operator site will be blocked by the Lac repressor, and hence the level of transcription is, or in other words we can say there will be low level of transcription because the operator site is blocked by the Lac repressor due to absence of the inducer lactose. Now in the second case, you can see the level of glucose is high, whereas the level of cyclic AMP is low. So in absence of cyclic AMP, the RNA polymerase cannot bind to promoter site efficiently, and second, the level of lactose is also low here. Thus the operator site is also blocked by the Lac repressor, as the repressor is not inhibited due to the absence of the inducer lactose. So there will be no transcription at all, or level of transcription is low. Number case 3, here the glucose is high, like the second case. The level of cyclic AMP is low, and due to this low level of cyclic AMP, the RNA polymerase will not bind to the promoter efficiently. But here the level of lactose is high, and this high level of lactose will inhibit the Lac repressor and inhibits its binding to the operator site. So the operator site will remain free, but since the RNA polymerase is not binding to promoter due to the lack of cyclic AMP, so again the level of transcription will be low. Now the case 4, here both the conditions are fulfilled. The level of glucose is low and the level of cyclic AMP is high. So the RNA polymerase will bind to the promoter efficiently, and the second, the level of lactose is high, that is the inducer is present. It will inhibit the Lac repressor and thus prevents it binding to the operator site. So operator site is also free, and RNA polymerase is also present and bind to the promoter site. So there will be high level of transcription. So I think the interplay of these molecules CAP, cAMP and glucose is now very clear to us. The level of transcription is only high when both the cyclic AMP level is high and the level of lactose, that is the inducer, is also high. It is interesting to know that bacteria can utilize both glucose as well as lactose. If a bacteria is supplied with both lactose and glucose, the bacteria will first utilize its first preferred source of carbon, that is glucose, and once it utilized its glucose supplement from the environment, it then start utilizing the second preferred carbon source, that is lactose. Because of this, a unique pattern of growth called dioxic growth is observed in bacteria. For more details, you can check my video on dioxic growth. For that, click on the I button. Now let's move to the effects of mutation in the different genes of the Lac Operon. The mutation can be broadly categorized into two types. One is called constitutive mutation. What is constitutive mutation? In case of constitutive mutation, the operon will remain turned on always, whether lactose is present or not, but the operon will remain turned on even in absence of inducer, that is lactose. Means here the role of lactose is neglected. Operon will always remain turned on. And the second one is uninducible, means here the bacteria cannot turn on its operon even in presence of lactose or the other favorable conditions like CAP is present, cyclic AMP is present, but the bacteria cannot turn on its operon if the mutation is an uninducible one. Apart from this, the mutation can be classified into dominant and recessive also. In case of dominant mutation, the effect of mutation or the mutant can be seen in presence of wild type or normal gene also. Whereas in case of recessive mutation, the effect of mutation cannot be seen if the normal or wild type gene is present along with it. It can be also cis or trans. In case of cis mutation, the mutation affects only the expression of genes on the same DNA, whereas in case of trans, the mutation affects the expression of genes on different DNA or chromosomes. Now let's see what are the different kinds of mutation. So first one is the Lac I negative. Here the repressor gene or regulator gene is mutated. If the regulator gene is mutated, what will be the consequence? It either do not synthesize repressor protein since the regulator or repressor gene is mutated, so the repressor protein will not be synthesized. And if the synthesis of repressor protein will occur, then it cannot bind to operator. And this is a trans, recessive and constitutive type of mutation. The meaning of this term I have already explained. The second is Lac IS mutation. In case of this, the repressor cannot bind to inducer. Means from the regulator gene, repressor protein will be synthesized, but it cannot bind to the inducer, that is allolactose. It cannot bind to allolactose. Third one is Lac O, that is mutation occurs in the operator gene. If mutation occur in operator region, the repressor cannot bind to operator. Means here the repressor protein is normal, but due to mutation in the operator region, the repressor protein cannot bind to operator. And the last one is Lac P negative. Here the promoter region is mutated. If the promoter region is mutated, the RNA polymerase cannot bind to promoter. Okay, let's understand the things through some examples. In the case 1, we can see here the I gene is normal, O gene is also normal, and the Z gene is also normal. So it is a normal operon system. So in case of normal operon system, in presence of lactose, the operon will remain turned on because the lactose will inhibit the repressor protein and the operator site will remain free, and the RNA polymerase will pass over to the structural gene and transcribe the structural gene Z. But in absence of lactose, there is nobody to inhibit the repressor protein and it will block the operator site. So the transcription will not occur. So this is a normal inducible operon system, because in presence of lactose, which is the inducer, the operon remains turned on. Now move to case 2. Here we can see the regulator gene is mutated. If the regulator gene is mutated, the repressor protein will not be synthesized. If there is no repressor protein, then there is nobody to block the operator site. So the operon system will remain turned on always, because there is nobody to block the operator site. The RNA polymerase can pass through the operator site over the structural gene and perform their transcription. So there is no matter whether lactose is present or not, the operon will remain turned on, and hence it is called constitutive, because in presence of lactose it will remain turned on, in absence of lactose also it will remain turned on. Now move to case 3. Here the operator gene is mutated. So if the operator gene is mutated, then what happens? You can see here, if the operator gene is mutated, the repressor cannot bind to operator. So if the repressor cannot bind to operator, then the operator site will remain free. If the operator site will remain free, then the RNA polymerase can easily pass over the operator site and perform the transcription of structural gene. So here also the operon will remain turned on if the lactose is present or if the lactose is absent, because the repressor protein cannot bind to operator site, because the operator site is mutated here. So this is also a constitutive operon, as the operon remain turned on in absence of lactose also. Now some other example. Here we can see in the first chromosome this is a normal one, but in the second one the I gene is mutated. Now what type of mutation in I gene? Let's see here, it is a recessive type of mutation. Means if the wild type gene is present, then the effect of mutation cannot be seen. So this mutation is a recessive one, and we have an wild type gene here. So we will see the phenotype of this wild type gene, that is I positive, and as a result, so this will behave like a normal operon system. In presence of lactose, the operon will remain turned on, and in absence of lactose, the operon will remain turned off. So this will behave like a normal inducible operon system. Now in case 2, here the mutation in the first chromosome, the mutation is in the operator gene, and the second one is the normal. Now what type of mutation is in the operator? Let's see here. If mutation occurs in operator, then it is a dominant type of mutation. Means the effect of mutation can be seen in presence of wild type also. In presence of wild type also. So if the mutation occurs in operator, the repressor protein will not be able to bind to the operator. The operon will remain turned on in absence of lactose also, in presence of lactose also. So this will behave like an constitutive operon system, because here the operator site is mutated and the repressor protein will not be able to bind to the operator site, and thus the RNA polymerase can pass over the operator site to the structural genes to perform their transcription, always, even in absence of lactose. So this is all about the different types of mutation and their consequences. So at the end, let's check once again what we have understand in this video. First, cAMP and CAP. CAP is an important activator of Lac Operon, as without CAP, the RNA polymerase cannot bind to the promoter more efficiently. However, the CAP also depends on another molecule, cAMP. So first, cAMP and CAP form a complex, and this complex will bind to the CAP binding site located in the upstream region of promoter, and thereby induces the binding of RNAP to the promoter. Thus, for strong induction or activation of Lac Operon or turning on the Lac Operon, both cAMP and CAP must be present. Number 2, what is the role of glucose? We know that glucose and cAMP are inversely proportional. If there is a high level of glucose, then there will be a low level of cyclic AMP. And if there will be a low level of cyclic AMP, it directly affects the binding of CAP to the CAP binding site and hence transcription. Thus, for strong induction, the level of glucose must be low so that a high level of cAMP can be maintained for its binding to CAP to form CAP-cAMP complex. And number 3, we have also learned about different kinds of mutation and their consequences in the activation of Lac Operon. We have learned about the uninducible operon or mutation. In uninducible mutation, the operon will never be, or we cannot, or bacteria cannot turn on the operon even if the conditions are fulfilled. Means CAP is present, cyclic AMP is present, and also the lactose is also present. Whereas in case of constitutive mutation, the operon remains turned on even if the allolactose is absent. And apart from this, the mutation can also be classified into dominant as well as recessive. In case of dominant mutation, the effect of mutation can be seen in presence of wild type gene or genotype. Whereas in case of recessive mutation, the effects of mutation cannot be seen if the wild type gene is also present. Similarly, it can be also classified into cis and trans-acting mutation. So I think I have covered all the contents like the effects of cAMP, glucose and mutation. Thank you for watching.